Okay, so for this topic, we're going to need to have page 10 and 11 of our digestion and metabolism booklet open. So page 10 and 11. Just pause until you get to the right page. So what we're learning about in this section is why proteins are important to our bodies. We're learning about what enzymes actually are and how they work, and factors that affect how fast an enzyme works. We'll not worry about some of these other points today. So let's think about proteins. We've talked already and said that proteins are important for, I think we said growth and repair, which to be honest is a bit of a key stage 3 answer. We need to up it a little bit to GCSE level. So we've got some notes in our booklet to help us. We shouldn't need to do lots of writing here. So proteins can be used for structural reasons within our body. They can be used for strengthening things like muscle. We know muscle could contract. So if you have a look at this animation, this is like, when you get to A-level biology, you'll learn about how muscles contract, but they're made of these long protein fibres that kind of fit together and pull over each other. Some hormones are made of protein, not all of them. We talked about some hormones are made of fat, but some hormones are made of protein. They act as little chemical messages, moving from one part of the body to another. This is a little cartoon showing how hormones work. So the little red squares are the hormones, and they fit together with little receptors on the outside of your cells, passing messages onto that cell. Proteins can be antibodies. You might have heard about these a bit more recently in the news. Antibodies are little chemicals that attach to bacteria and viruses. They're made by your white blood cells. And so the antibody in this picture that's just popped up is green, and the pathogen, the virus or the bacteria, is red. And can you see the antibody fits together perfectly with the little spikes on the outsides of the pathogen? And also proteins could be catalysts in your body, enzymes. So they're chemicals that speed up reactions. So this little yellow thing that's popped up here is a little enzyme, and you can see that it's got a really special shape bit cut out of it. So what can we spot about all these proteins that we've talked about? What's in common with how they all work? Maybe you can spot, pause if you need a bit of thinking time, but maybe you can spot the idea that they all involve like jigsaw pieces fitting together. So like the muscle, you can't really see it on this diagram, but the muscle fibres fit together and slide over each other really nicely. The hormones fit together perfectly with the little receptors on the outside of your cells. The antibodies fit together perfectly with the little spikes on the outsides of the pathogens. And we'll learn today about how enzymes fit together perfectly with the thing that they're trying to break down. So it's all about fit. So proteins basically have to have, if they're going to work in your body, they have to have a really, really specific shape. If you change the shape, they're not going to do their job anymore properly because they're not going to fit with the right jigsaw piece. So what are proteins made of? Well, we learnt last lesson that proteins are long chains of amino acids, and in your body you have about 20 different amino acids that are used to build up a huge variety of proteins. So if you think about your body, if you take out the water, obviously that's an important chemical, but the vast majority of the stuff that's left behind, if you were to shrivel yourself up without the water, is protein. Your muscles, your hair, your nails, lots of parts of your blood, your bone marrow, etc. is all made of protein. And we've got these 20 different building blocks that we can use to build all the different proteins in your body. So it's just like having 20 different size Lego bricks. You can put them together in a long straight chain, you can put them together in a block, you can do all kinds of stuff with them, and you can make lots of different shapes. It's the same in your body. You've got these 20 building blocks, but you can put them together in any order and make yourself lots and lots of different shaped proteins. And we know that the shape of the protein is really important to do its job. So they're all folded up. Once you make these big long chains, I mean they could be hundreds of amino acids long, you fold them together to make a really specific shape, and that allows that protein to do its job. We'll learn about in year 11. We don't need to really read this, worry about it too much at the moment, but we might remember from in fact we revised this recently, the idea that ribosomes make protein. So your DNA codes for protein, your ribosomes will kind of read that code and they build together the protein. So this picture is just showing you eat lots of protein in your diet. You can see the balls at the bottom. I don't know if I can annotate this. See these balls at the bottom here are like a protein. Lots and lots of amino acids in a chain. You digest them and you absorb them into your blood, these single amino acids. Can you remember the name of the enzyme that does this chopping? See if you can pause and say it. It's protease enzyme. So these little amino acids will go into your cells, and your cell, your ribosome specifically, will join them all together in a chain to build a protein that's useful in your body. I'm going to put this video separate because it's not going to play right through the PowerPoint. But if you pause this video, go into the next one, and then come back to this. So let's think about one type of protein specifically, enzymes. So enzymes are important in so many different chemical reactions that go on in your body. You've got lots and lots and lots of chemical reactions in your body, and that all together we call that your metabolism. But we've been talking specifically about digestive enzymes, haven't we, the last couple of lessons? So enzymes are biological catalysts. They increase the rate of chemical reactions in all living cells. Enzymes are proteins, or a type of protein, and they're very specific. This is an important word. So each enzyme will only catalyze a particular reaction. So for example, protease will only catalyze the reaction of proteins to amino acids. Lipase enzyme will only catalyze the breakdown of lipids to fatty acids and glycerol. Amylase enzyme will only catalyze the reaction of starch being broken down to sugars. And that's because they've got a very special shape called an active site. We'll find a picture of that here. You've got these notes in your book, but you can add whatever extra detail to your booklet you want. So these big blue blobs, the light blue ones, are enzymes. Remember, they're made of protein. If I was to do a super zoom in, I would actually find that they're made of huge chains of amino acids, but I'm not drawing all that in here. Can we get rid of all this? No idea. And this enzyme has got this very specific shape region here called the active site. It's a really, really specific shape. So the substrate, this means the thing that's being broken down, it fits perfectly into the active site. So let's say, for instance, let's give an example. Let's say the enzyme was amylase. What's the name of the substrate that fits into amylase's active site perfectly? You remember? Have a look at your notes from last lesson if you're not sure. The answer is starch. If the enzyme was protease, what would the name of the substrate be? What would the name of the thing that gets broken down by protease be? It would be protein. If the enzyme was lipase, what's the name of the substrate that lipase breaks down? It would be lipids. So each enzyme has a specific substrate. That word is super important. And the substrate fits in the active site just like a lock and a key fit together. So we call it the lock and key theory. When they join together, we call it an enzyme substrate complex. And then the enzyme will do its work, it will break some bonds, and it will release some products. So for example, let's just recap these ones again. If the enzyme is amylase, what is the substrate? The substrate is starch. And what would the product be if starch was being broken down? Do you remember? It would be sugars. If the enzyme is protease, what is the name of the substrate? The substrate would be proteins. And what would the products be? They would be amino acids. See if you can be answering these questions as you go through. You can keep rewatching the video as many times as you need to check yourself. If the enzyme was lipase, what would the substrate be? The substrate would be lipids. This is a bit trickier. What would it get broken down to? Fatty acids and glycerol. Okay, so we've got this basic idea called the lock and key theory. You've got some pictures in your booklet on page 10 to show you what's going on. Now let's think about what can happen to this enzyme at different temperatures and different pHs. Well, if you imagine this active site has got a really specific shape, hasn't it? If you heat an enzyme up to maybe above 40 degrees, we know that if we heat up molecules, the little atoms in the molecules start to vibrate a little bit more. And if you heat them up too hot, they vibrate too much and they change the shape of the active site. Maybe it goes from being this shape to being more like this shape. Now, what's going to be the problem there if you change the shape of the active site to something that looks like this instead of its original shape? Why is that going to be a problem? Because the substrate no longer fits in the active site perfectly. So you don't form enzyme substrate complexes. Therefore, you don't get any products being formed. So effectively, if you change the shape of the active site like that, you've permanently damaged that enzyme. And we say that the enzyme has become denatured. There's this word. Enzymes are denatured by high temperatures. And the reason for that is the active site changes shape. And that is a permanent problem. So again, you can add whatever extras you need to this set of notes to help you. So this graph on the left here shows the effect of temperature on an enzyme activity. So temperature is going from freezing up to, the body temperature is about here, isn't it? 37 degrees C. So at really cold temperatures, you have a really low rate of enzyme activity. And this is because for an enzyme to work, if you think back to this situation here, the substrate and the enzyme have to bump into each other. The enzymes are not in fixed positions usually. They're bumping about in the cytoplasm. So imagine you've got a cell and you've got your nucleus. You'd have loads and loads of enzymes. And those enzymes will be just bumping into substrates the whole time. Some enzymes are sent out of the cell actually, so they might be just floating about in your small intestine tubes. But regardless, for an enzyme and a substrate to join together, they've got to bump into each other. And we know when molecules are really, really cold, they don't move much. They have very low kinetic energy. So that might be worth putting in here. Low kinetic energy. And so when you've got low kinetic energy of those particles, you've got fewer collisions between enzyme and substrate. Sorry about my writing. But as you heat the enzyme up a little bit more, you get more kinetic energy, and so more collisions between the enzyme and the substrate. You'll reach a point up here. This is called the enzyme's optimum temperature. In our body, our enzymes have an optimum temperature of about 37 degrees C. That's why we keep ourselves at 37 degrees C. They will work a little bit, they work equally well at maybe about 38, 39, 40, but anywhere past that, we start to get a problem. And can you see the enzyme rate decreases with hotter temperatures? And this is that idea that the active site permanently changes shape. And that means that there are no successful collisions, I spelled that wrong, sorry, between the enzyme and the substrate. So the enzyme can no longer fit together with the substrate. So no product is formed. If you get a specific exam question that doesn't just ask you about enzymes in general, maybe it asks you about amylase, rather than saying no product is formed, be specific, no sugar is formed. If the question's about amylase and it says the active site of the amylase enzyme permanently changes shape, therefore no successful collisions between the amylase and starch. Like, make these into specific words if you know which enzyme you're talking about. So this is permanent. This is called the enzyme being denatured at this point, when it's really, really hot. pH has a similar issue on the go. So along the x-axis here, we've got the different pHs and the rate of enzyme activity on the y-axis. Every enzyme has got its own optimum pH, and it's not fair to say that they all have an optimum of about 7 or 8. Can you think of a part of your body where the enzymes have to have an optimum pH that's really acidic? So that would be the stomach. So maybe the the protease enzyme that's produced in the stomach would have an optimum pH really, really low because it's working in hydrochloric acid. So excess, this side here, excess acid or excess alkali on this side denatures enzymes. I'm hoping you can kind of read my writing and put some notes on your thing. And again, that's the idea that the active site changes shape. So the acid, it's not the temperature this time, it's changing the shape of the active site, it's the acid or alkali. So active site changes shape, so the substrate no longer fits. Same with this one. Same bullet points here. So excess alkali denatures enzymes, the active site changes shape, the substrate no longer fits. When we do some exam questions on this in a bit, you'll see the language that they want you to use in your exam. Okay, so that's the theory done. Have a look at Firefly for your next couple of jobs.